Welcome, grade 8 students. How are you? Today we are going to continue our lesson about the specific immune response. Let's memorize in case of invading microbes pathogen. There are two ways of defense: innate immunity and acquired immunity. Innate with rapid response to a broad range of microbes, and acquired if there is a slower response to a specific microbe. For example, in case of innate external defense and internal defense, external by skin mucous membrane secretions, and internal by phagocytic antimicrobial protein inflammatory response and natural killer cells. In case of acquired immunity, humoral response by antibody and cell-mediated response by cytotoxic lymphocyte. Modalities of specific immune response: cell-mediated immune response, this is by T lymphocyte, and the humoral immune response by B lymphocyte. The natural killer cells recognize surface molecules on abnormal cells in case of cancerous or virus infected cells. Acquired immunity, the immune response, is highly specific, produces antibody in response to specific antigens. Antigens may be molecules on bacteria, virus, protozoa, worms, transplanted organs. Both B and T lymphocytes have receptors on membrane that recognizes different antigens. B cells, mature in bone, produce antibodies. Antibodies flow through the blood, recognizing and attaching to antigen, while T cells, mature in thymus, do not produce antibody. Cytotoxic T cells require cell-cell contact to destroy pathogen. Both B and T cells can produce memory cells. Look at this figure. This is the production from stem cells in the bone marrow of lymphocytes of immature, which means that they are non-mature. T lymphocyte go to at the level of thymus, uh, which take maturation and forms antigen receptor. This is the T cell. And B cell in the bone marrow, it matures up antigen receptor. This is the B cell. Both, this is the cell mediated affected by T cell, and this is the humoral affected by B cell. Both of them via blood go to the infected part in the lymph node, the spleen, and other lymphatic organs. Final maturation of B and T cells in lymphatic organs. Primary immune response: T cells are selected, B cells are selected. What is the selection? Antibody producing plasma cells are produced, person feels ill. While these cells are produced, symptoms diminish as these cells do their job. Secondary immune response: this is the primary immune response, and this is the secondary immune response. There are two kinds of immune response. In case of specific immunity, look at the concentration of antibody. It starts with null. It increases first in primary immune response to reach a peak, a moderate peak, after initial exposure to the antigen. After the second exposure to the antigen, a secondary immune response, the amount of antibody increases in a large amount. Response is much faster. Memory cells are activated. They tend to have stronger response than the primary. This is the primary immune response. B cells with different antigen receptors. This is the antigen receptor. Antigen molecules. This is the molecules. First exposure to antigen, there is amplification, cell activation, growth, division, and differentiation. Antibody molecules are secreted. This the first clone endoplasm by endoplasmic reticulum, plasma effector cells secreting antibodies. And in the same time, the memory cells are formed. In secondary immune response may occur long after primary immune response. This is the second clone. Antigen molecules look at the second exposure to the same antigen. There are amplification and formation of memory cells. This is a more clear figure for primary and secondary immune response. Antibody concentration. We have two antigens, X and Y. In the first exposure to antigen X, a primary immune response to antigen X or formation of antibody anti-X. Antibodies, the amount of antibody increases moderately. After the second exposure, it increases in high amount. This is the secondary immune response to antigen X. While the first exposure to antibody Y, it increases. This is the primary with respect to the antigen Y. T cells work by directly binding to infect cells and then destroying them. This is the mode of action of T lymphocyte. Cytotoxic T cell bind to infected cell. This is the self and non-self complex. This is the infected cell, and this is the cytotoxic T cell, which has perforin molecule. We will see the role of perforin. Perforin makes holes in infected cells membrane and enzyme enters. It makes a hole and enzyme enters inside the infected cell. Infected cell is destroyed. This is the end of our video. Thank you for watching and goodbye.